Hello and welcome everyone to the last session in our GenScript webinar series this year. My name is Keshav Vasantavada and I'm a marketing specialist for protein services at GenScript. Just a few words about me. Prior to joining GenScript, I worked for 7 years in a big pharmaceutical company in their core facility, performing cloning, expression, and purification of a variety of disease targets. I have hands-on experience with different expression systems, but my work has predominantly been with E. coli. Among other things, I have experience in the development of novel fusion partner technology for recombinant soluble protein production and the implementation of automation and liquid handling systems in performing small-scale expression studies in a high-throughput fashion. And so in line with my background, the topic of my talk today is fusion partners for recombinant soluble protein production in E. coli. So as scientists in the life sciences area, we all realize at some level the importance of recombinant soluble proteins in discovery research, right? So as you know, E. coli is a popular choice for the production of these proteins. And so in my earlier webinar, which was aired in around May, June this year, I had reviewed what we found were the most important optimization parameters that really influence soluble protein expression in E. coli. I hope that some of you joining us today had a chance to listen into that webinar. If you didn't, I would certainly recommend that you take some time to review it. So in today's webinar, I'm going to discuss a fusion partner strategy for the production of soluble proteins in E. coli. So while there are various ways to optimize expression, fusion partner based approach can be really useful. So in this webinar, I'll try to review the practical aspects of a fusion partner strategy. And in doing so, we leverage the collective experience of our production team that has generated over 5,300 batches of really high quality pure recombinant protein so far. And so I really hope that this session will help you build a better E. coli expression strategy while avoiding some common pitfalls in your experimental design. So if you have any questions during the presentation, you can submit them using the chat box. I'll do my best to answer some questions at the end of the session. And if you're unable to sit through this presentation, you can check back on our website, www.genscript.com, to view this archived webinar and links or downloads of any associated material that we present today. So quickly, the overview. This presentation is broadly divided into 5 topics and the conclusion. I'll do a brief introduction about GenScript before introducing the topic, and then I'll take a few minutes to talk about some of our important E. coli expression services. And then we'll look at the challenges with in working with an E. coli system and ways to address some of those challenges. At this point, we'll discuss more about protein solubility and the factors that really determine protein solubility. We can take a look at a typical E. coli protein expression workflow, and then look at some of the key aspects of E. coli expression, and what you need to consider before embarking on a protein expression project. We can focus on concerns and aspects of expression, E. coli expression, and look at the solutions that GenScript has come up with to address some of the challenges. Then I'll list out what we think are some of the most important optimization factors that contribute to recombinant protein solubility, really with an emphasis on fusion partners. And then we can take a look at 2 case studies, review 4 important fusion partners, and discuss the pros and cons of a fusion partner based approach. And then I'll recommend a broad strategy before we conclude. So about GenScript, right? Headquartered in Piscataway, New Jersey, GenScript is the world's leading biology research service company with gene synthesis, peptide, protein, antibody, cell line, and discovery biology service capabilities. We are internationally recognized as a leading contract research organization specializing in biological research and early phase drug discovery services. We provide services and products to scientists in 86 countries really around the world, and we built the best in class capacity and capability for biological research services encompassing gene synthesis, molecular biology, peptide synthesis, custom antibodies, protein expression, antibody and protein engineering, and in vitro and in vivo pharmacology. Now all this with the goal to make research easy. Now more than 10,000 peer-reviewed journal articles have cited GenScript's services and products, making GenScript the most frequently cited biology contract research organization in the world. So we know that recombinant protein production is an essential step for the functional characterization and structure determination of proteins. And while a variety of hosts are available for this purpose, the gram-negative bacteria E. coli is often considered the quote workhorse of researchers, right? And hence my talk today will focus on recombinant protein production in E. coli. So in doing so, we are going to leverage the collective experience of our production team. As I said earlier, they have shipped more than 5,300 batches of pure protein so far. So one of the most important things you need to consider before embarking on your project is the host, right? So traditionally recombinant proteins have been made in hosts like bacteria, insect cells, yeast cells, or in cells of a mammalian origin. There are also kits available that would allow you to make protein in vitro using a cell-free translation system. Now given this kind of choice, how do you select which expression system to use? So while there are various factors that go into that decision making process, the nature of the target protein, its solubility properties, its functionality downstream, the yield requirements that you desire, user experience, and the speed of generation. These are some of the most important factors that govern the decision. And in this regard, E. coli is often the number one choice of researchers around the world. And this statement is attested by the number of entries in the protein data bank by expression system as a percentage of total number of chains with an identifiable expression system. And this data was collected in earlier this year in April. But the data still holds true. I'm sure if you were to pull this up today, the percentages wouldn't change much. So once you decide to go with E. coli, right, the next logical question that comes to mind is, how am I going to make soluble protein? And again, this depends partly on the application and partly on the target protein itself. A lot of the times soluble protein can be made during the small scale optimization step, but it's not possible to reproduce that and scale up. It doesn't work all the time. So sometimes you need to think early on in case you're trying a chaperone co-expression strategy or a fusion partner approach, because that could require some target manipulation early on, or it could require the use of multiple constructs. So on the topic of E. coli, I'll just take a few moments here to introduce you to some of our E. coli expression services that will help you reach your goals faster should you choose to partner with us. Now we have the backpower guaranteed bacterial protein expression package, which is the table on the top. Now this package offers a comprehensive panel of options for your target protein production in E. coli, with guaranteed protein amounts, guaranteed purity, endotoxin levels, and the option of producing tag-free proteins that meets a variety of your needs. All you have to do is just provide the protein sequence, and we'll deliver the protein either tagged or tag-free at the amount and purity that you specify. Now this is a great value package because it's zero risk. By the way, you can also get an instant online quote for this particular service within seconds from our website. Backpower custom services, the table in the bottom. GenScript does not use a one-size-fits-all approach to protein production, because every protein is different. Every step of the process is treated uniquely, right from codon optimization to gene synthesis to small-scale testing, scale-up, and purification and characterization. So what's the difference, from our guaranteed service? So our custom services are best for higher difficulty targets. So for example, if you have membrane proteins, toxic proteins, ion channels, custom is the way to go. Also, if you have any special requirements, like you want to send your own DNA samples or your own protocol, the custom packages are for you. Fast gene to protein, the table on the top. With the growing pace in research, more and more projects need proteins really quickly. As such, the faster production time for high quality proteins is in demand, and it can add significant value to your research. To address this unmet need, we launched the Fast Gene to Protein service. And just so you know, this is the fastest protein production service in the industry. It takes you from sequence to purified protein in just 4 weeks. Now, obviously, this is ideal when you have a time-sensitive project. So frag power, the table in the bottom. So experts suggest that antibody fragments are going to be the next important class of protein-based therapeutics, right after monoclonal antibodies, right? So fragments in particular, the scFvs, the Fabs, and variable heavy chains, these retain full antigen binding capacity and superior properties for research, diagnostic, and even therapeutic applications. And because of this rising need for fragments, we launched the Frag Power Guaranteed service that provides high quality fragments at very competitive prices and timelines. I hope that you will take some time to learn about these and our other protein services after this webinar. So going back to the topic. So we know that E. coli is a top choice, right, for protein expression, but what makes it favorable and what are some of the disadvantages? A combination of simple genetics, ease of culturing, scalability, fast expression, and high yields makes it really attractive, while the lack of an efficient post-translational machinery, codon usage bias, and difficulty in producing high molecular weight proteins makes it disadvantageous. But one of the biggest evils in E. coli expression, which is nearly as bad as low expression or no expression, is insoluble expression. As all of you will be familiar with, this is a phenomenon when overexpression results in the formation of insoluble aggregates called inclusion bodies. Let's recap the possibilities in an E. coli expression project. So here are two of the most likely scenarios you encounter when expressing your target in E. coli. Right? Number one, which is the most favorable outcome, would be that you're able to overexpress your protein in a soluble fashion in the bacterial cytoplasm and everyone is happy. Right? Or you could try overexpression, but due to anomalies in protein folding, the protein ends up as insoluble aggregates called inclusion bodies. And when that happens, you could try a very cumbersome process called in vitro protein refolding and still get the necessary outcome. Or you could try optimization at the process or molecular levels. So at the process level, you could adopt methods that do not require target engineering. For example, you could play around with the expression conditions, like trying out different strains or adjusting the growth induction conditions, changing the media, buffers, chaperone co-expression, and even try to improve the cell lysis conditions. The alternative would be to manipulate your target protein by eliminating any undesirable elements within a sequence. So you could create truncations or mutate certain amino acids to make the protein more soluble. And you could also adopt a rational site-directed mutagenesis approach based on structure information. Or you could take a directed evolution approach and screen for the solubility of random point mutants, deletions, and fragments. So better, easier approach would be to adopt a fusion partner strategy and use a solubilizing partner upstream of your target protein to make it more soluble. So really, why is target protein insolubility such a big deal, right? Just to give you a perspective of how bad the insolubility problem is, I have these statistics from the Northeast Structure Genomics Consortium's website that indicates that out of the approximately 24,000 constructs that were expression tested, nearly 43% were found to be in the insoluble fraction. So as you can see, target protein insolubility is indeed a significant problem. And speaking of insolubility brings me to state the premise of most protein production projects, which is the generation of high yields of soluble protein in a cost-effective and expeditious manner is the single most important determinant of a successful protein production campaign. That is, all these variables in your expression need to go through a carefully crafted funnel of optimization in order to yield soluble protein. Now I know there will be skeptics out there who are listening who will tend to disagree with me, and sure enough, with reason. Solubility need not be the be all and end all of a protein expression campaign, but for most purposes protein solubility often becomes a direct or indirect measure of protein quality. So what really is the relationship between protein solubility and its functionality, right? So for a long time there was a very straightforward relationship between the solubility of a protein and its functional behavior. Now, for example, if a protein was soluble, there was high likelihood that it would behave as expected in a functional assay, and vice versa. If a protein performed well in a functional assay, it was believed to be soluble. If a protein was insoluble, that spelled doom for the project, because that meant that the insoluble protein would be entirely useless. And so people would go about devising other ways to generate soluble protein, including refolding from inclusion bodies. But this scenario has been changing gradually. Many studies have shown that even properly folded and functional proteins often aggregate as inclusion bodies. And so an important fraction of functional proteins also occur in the insoluble fraction. Also, the occurrence of insoluble but functional protein indicates that solubility and the conformational quality of your recombinant protein, they are both exclusive parameters, and they both can be engineered independently. And so many scientists actually believe that the bioactivity, rather than just the soluble nature of a protein, reveals the true quality, the true conformational quality of a protein, and hence its true potential. So if that really is the case, you could wonder why all the hype about soluble protein expression, right? And that's a true concern. Now that's because most of the time, for lack of another easier predictor, solubility is often used as the best indicator of a protein's functional behavior. And also, since there is no way to really make functional protein, solubility is the closest indicator to functionality. I hope that made some sense. Now, having established that, then let's take a look at the factors that really determine protein solubility. Now, it has been shown that two parameters are really critical in making the distinction between insoluble protein versus soluble protein. So what are they? Number one, charge. The charge on the protein, so what does this mean? So the charge meaning the difference in the numbers of aspartic acid and glutamic acid versus lysine and arginine residues. Okay. And number two, the turn forming residue content, which is the total number of asparagines, glycines, proline and serine residues. So let's take a closer look at these parameters. So really, why is charge important? We know that the functional groups of amino acids determine their charge at a given pH, right? So for example, at neutral pH, aspartic acid and glutamic acid, they carry a negative charge, and arginine and lysine, they carry positive charges, right? So the net charge of a protein molecule is the arithmetic average of all charges, right? So the protein net charge simply depends on the number, identity, and location of amino acids and the solvent pH. Because when the pH changes, the charges on all these amino acids change, right? So at a certain solvent pH, the protein net charge becomes 0. So as you know, that's the PI or isoelectric point, right? So for example, if you take a look at this schematic here, we have a protein for which the PI is around 8.5, right? So what happens when the pH of the solvent decreases, becomes more acidic? So as the pH drops, you know that more hydrogen ions are available in solution, and these ions bind to negative sites on amino acids, and the protein as a whole becomes positively charged. On the other hand, at basic pH, the protein becomes negatively charged as a whole. And when you have no net charge on a protein, the proteins start to interact among themselves instead of interacting with the surrounding water molecules. And so this really in turn leads to aggregation. So hence charged protein molecules tend to be more soluble than proteins with no net charge. So number 2, the turn forming residue. So the other important thing, turn forming residue, if you have more number of prolines, serines, glycines and asparaginase, the higher likelihood that your protein will be insoluble because of slower folding rates of your protein. And so if you can figure this out in advance, just quickly analyze the amino acid content of your protein, you can avoid rapid overexpression at high temperatures. You could instead perform induction at low temperatures. So again, quickly to summarize, more net charge on protein is good, more turn forming residues in protein is bad for solubility. Okay, so now let's take a look at what a typical protein expression workflow looks like. And while I show this as a sequential chain of events, you know that some of these steps work in parallel. So after a project has been initiated, the first logical step is literature review, right? Where you scour the web to see if there is a literature precedent for your target. That is, maybe it's been produced already by another group. If so, can you get the protocol and reproduce it? And again, while this is a necessary step, all bets are off because you know that reproducibility, in life sciences is not guaranteed. So in other words, what has worked for one group need not work for you, even with the same target. Now, with or without literature precedent, we move on to the next step, which is target analysis in engineering. It would be good to know if the target was expressed full length, which would be the best case scenario most of the time. An initial analysis will tell you if the target will be a challenge to produce. There are recombinant protein solubility prediction programs which can tell you the probability that your overexpressed target will be soluble in E. coli. Other considerations are, will you make tagged protein or will it be tagless? Will you place tag at the N terminus or C terminus? What's going to be the cleavage tag? We already reviewed the host consideration parameters earlier. So once you design the target, you begin the cloning scheme, right? So typically many researchers these days, they tend to adopt parallel strategies to facilitate high throughput cloning and expression. So for example, strategies like ligation independent cloning or recombination based cloning, these allow you to generate multiple expression constructs at once, which is a good approach, while ligation independent cloning is friendlier in the sense it adds minimum vector residues, and it's perceived as more friendly for crystallography. Recombination based cloning is also a very efficient method. Now these days many scientists actually have their genes designed, synthesized and subcloned into an expression vector directly, and GenScript is very good at doing that should you choose to outsource. So once the construct is ready, the next step is small scale expression evaluation optimization, with the last step being scale up and purification. And this teardrop symbol along this workflow symbolizes all the steps that GenScript can perform for you to support and accelerate research. So this is similar to an earlier slide where we look at a bacterial expression project more closely. So once you attempt expression and see that the protein is soluble at small scale, you could either archive the construct or scale up and purify it. On the other hand, if the protein does not express at all, we need to review the construct carefully to make sure there is nothing wrong with it. Right? For example, is the DNA sequence okay? Is there a frame shift mutation that results in no expression? Is the vector all right? Is the protein toxic? And if your protein is insoluble, we have our proprietary program called FoldArt. It's a technology that combines high hydrostatic pressure techniques, small molecule additives, and column refolding methods to yield pure protein with over 95% success rate. And we can also use potential to perform evaluation and optimization for you. And during expression optimization, we also recommend running a small scale test purification in order to get the most accurate report of your protein's behavior. So here I'll take a few moments to introduce you to our potential service. So what exactly is potential? It's a protein expression evaluation and optimization service that we offer. You can evaluate whether your target protein expresses in your chosen system. You can identify the best expression system for your target protein and identify the construct and conditions that give you the most robust soluble expression. The goal is to evaluate thoroughly before scaling up in order to save time and resources. At GenScript, we have a comprehensive one-stop service that includes gene synthesis, subcloning, protein expression, evaluation, and optimization, all the way to large-scale protein production and beyond. So there are essentially three different potential packages. We have the standard package that starts at $280, which is available for bacterial, mammalian, and insect cell expression. We have the silver package that starts at $1,440. That is available for E. coli system only. It allows you to identify the best expression condition with your chosen vector and bacterial strain. Now the potential gold package is an industry first high throughput expression and solubility optimization matrix that tests 48 different conditions to establish the ideal condition for your target protein. Now an optional but recommended add-on 1 liter purification step is also available with this service. Now this slide here depicts some of the parameters that impact protein solubility, and playing around with these factors could help you achieve your desired goal. So we feel that inducer, growth media, induction time, chaperones, vector and promoter choice, codon optimization, E. coli strain, temperature, and fusion partners, these are all important considerations. And since I discussed most of these aspects in my previous webinar in May and June, I'll skip the rest and go straight to the fusion partner topic. So this table here lists some of the popular affinity and or solubility tags. By the way, I should just clarify that in this webinar I've been using the terms tag and fusion partner interchangeably, so just so you know. So basically these tags are divided based on their utility as an affinity or purification tag versus solubility tag. Now some tags perform a dual role. They're both affinity and solubility tags. So as you'll know, one way to improve the solubility of a target protein is by fusing it downstream of a soluble fusion partner, and this in my opinion is the best way to produce soluble protein. Now of course each tag has its pros and cons, so we need to evaluate the experimental requirements carefully when we select a particular tag. And this table we list some commonly used tags. Just note that the ones in red font are those that I'll discuss a bit more in the next few slides. So how do you choose a tag? That depends on a variety of factors, right? For example, the cost of your affinity resin, its regenerative capacity, its binding capacity, the purification buffer conditions, etc. Right? And obviously expression levels are really important. So for example, solubility tags like GST, MBP, News A, they have really strong translational initiation signals that can drive high level expression. So you need to be careful at times, because if you need low level expressions, for example, if you're studying complexes, protein complexes, it would not be advisable to use one of those tags. Then it would be better to go with a more stringent epitope tag instead. Also, you need to decide whether the tag sits at the N terminus or the C terminus. While many on this list can be engineered at either end, some perform really well at one end over the other. So the advantage of having an N-terminal tag is that the construct can utilize the efficient translation initiation signals of the tag, right? Another advantage would be so that the tag can be removed more efficiently if it's at the N-terminus, because more proteases cut near the C-terminus of the recognition sites. So the cut is much more cleaner if your tag is located at the N-terminus. So about the his tag. So as you know, the his tag is quite universally used as an affinity purification tag, but just remember that the his tag is not as innocuous as some would think. It can affect expression levels, protein solubility, its bioactivity, stability, and even the structure, and overall function of your protein. Sometimes, not always. CBP is short for calmodulin binding peptide. It's a really short affinity tag that uses immobilized calmodulin cephalose for affinity purification. DSBA, it can increase solubility both in the cytoplasm and periplasm, and it also helps with disulfide bond formation. Flag tag. The flag tag functions really well, and it also introduces an enterokinase cleavage site. But remember that the flag resin can be a bit expensive. Strep tagged proteins bind really tightly to streptavidin columns and are eluted under physiological conditions, buffer conditions, so much more easy to use. The sumo tag is a really good solubility tag, and the advantage is that the sumo protease cleaves the tag with really high specificity, because it recognizes the tertiary structure of sumo and cleaves at the C terminus. So you can recover target protein in a very pure form, with no extra residues. And this slide here illustrates the value of a fusion partner approach in producing soluble protein. Now these are both Kumasi stain gels. On the left side image you see target protein A. It has a molecular weight of 30 kilodaltons. It's barely seen in the soluble fraction where the red arrow is, nothing there. But once I fuse a fusion partner upstream of it, it's a 15 kilodalton fusion partner. This makes the target protein really robustly soluble. So as you can see, lanes 1 and 2, the difference between no tag and with tag. And on the right side image you can see a 15 kilodalton highly insoluble target protein is made really soluble by tethering it downstream of a 45 kilodalton soluble fusion partner, as you can see in lanes 3 and 4. And by the way, both these fusion partners are novel proteins that are not on the list that we saw on the earlier table. So about 4 different tags. So GST tag, GST is an abundantly expressed 26 kilodalton tag isolated from this parasitic worm, Cystosoma japonicum. Typically GST is fused at the N-terminus of a target protein, and when fused at the C-terminus, GST is not very efficient. It doesn't mean you can't use it. You could still use it at the C-terminus as an affinity tag. Now GST has been used for recombinant expression in E. coli, mammalian cells, insect cells, and even in yeast. So in that sense it's really versatile. GST binds to immobilized glutathione in a resin, and this you could use this property for affinity purification. So to elute, you basically use reduced glutathione, and resins used for GST purification are relatively cheap, and they have really high binding capacity, and they can also be regenerated and reused multiple times. So all in all, a pretty good tag. But the downside is that the GST has to be properly folded in order to bind glutathione, and so the fusion protein needs to be soluble and in non-denaturing conditions for you to be able to purify it efficiently. Excuse me. Detection is easy because there are many commercially available anti-GST antibodies. A disadvantage though is that GST forms a homodimer in solution, so that could be a poor choice if you're trying to tag oligomeric proteins. And also the kinetics of GST binding to glutathione resin and its elution, they are really slow. So GST fusion proteins need to be loaded, washed and eluted at really slow flow rates. You can't do it quickly like with the his tag. The NewSA tag, the N utilizing substance A, also known as NewSA, is a really large tag, and this was chosen as a potential tag, since it ranked among the most soluble E. coli proteins based on a statistical solubility model. In some large-scale screening tests, NewSA was reported to perform equally or even better than MBP as a solubility tag. But that doesn't mean we can conclude that the NewSA is better than the MBP, only because the results vary for individual proteins. Now NewSA does not double up as an affinity tag, so NewSA fusion proteins are typically used in conjunction with another affinity tag, usually the his tag. Thioredoxin is a 12 kilodalton thermostable intracellular protein. It's ubiquitously found in all living organisms, all the way from bacteria to higher mammals. It functions in cellular proliferation, redox signaling, and in the inhibition of apoptosis. Thioredoxin is highly soluble even when you overexpress up to 40% of the total cellular protein, and it's very useful as a tag in avoiding inclusion body formation. Now various studies have indicated that thioredoxin is more effective when you place it at the N-terminal end. And one of the advantages with thioredoxin is that it accumulates at cytoplasmic membrane adhesion sites, which allows for a simple osmotic shock or freeze-thaw treatment in order to release the protein. You don't have to go through a complex procedure in order to lyse the cells. It's very simple. Again, like with the NewSA, this one does not perform as an affinity tag, so you need an additional affinity tag, like the his tag, in order to use this for purification. MBP, the MBP is quite robust. It's been widely used. It's an approximately 43 kilodalton tag. It naturally occurs in several bacteria, and primarily in E. coli. It's encoded by the mol-E gene, which is responsible for the uptake, breakdown, and transport of maltodextrin. carbohydrate. Um, Originally this was developed as a protein expression tag in the 1980s, and it's known to significantly enhance the solubility of a variety of target proteins. Now this tag is typically used at the N-terminus, but it's also effective when placed at the C-terminal end. The large size of this tag puts a really heavy metabolic load on the host, but in some high throughput tests MBP was actually ranked as one of the best tags for making soluble fusions. So how does this work? The exact mechanism of protein solubility enhancement by MBP is not entirely clear. It's known to stabilize and protect its downstream passenger protein from proteolytic degradation, both during and after protein synthesis. Now cytoplasmic yields of MBP fused proteins are also usually higher because the fusion protein provides reliable contexts for efficient translation initiation, and it's also proposed that the MBP tag might act as a chaperone by interacting with solvent exposed hotspots on the target surface, which stabilizes the otherwise insoluble passenger protein. Now commercial vectors for MBP tagging with a variety of cleavage sites are available, both for cytoplasmic and periplasmic expression. Cross-linked amylose resin is used to bind MBP tagged proteins, and the fusion protein is typically eluted under gentle conditions using maltose. So that's about 4 different major tags being used in labs around the world. So what's the downside of using a fusion partner approach? While this strategy is effective for the production of soluble proteins, obviously it has its drawbacks. Firstly, most of the time the target protein needs to be cleaved off because they are too large, and they might interfere with the target's natural function, right? Of course, some groups have shown that you can generate crystal structures and perform functional assays with even with full-length fusion proteins without really having to cleave off the tag. Number 2, now removal of a C-terminal tag can be tricky because oftentimes you have extraneous amino acids left over from the protease cleavage site. And then again, cleavage itself can be an issue due to steric hindrance. And this can be resolved through the addition of a linker region between the proteolytic cleavage site and the fusion partner. And in some cases, a certain resin can be fragile and relatively expensive. For example, amylose resin can be really fragile. Flag resin is really expensive. And then what else? Target protein precipitation post cleavage. Now this can be an issue which can be often addressed by optimizing the protein purification conditions. And some fusion proteins do not bind very efficiently to the resin, and even when they do, yields can be an issue. And this can also be occasionally addressed by optimizing the protein purification conditions or by changing your affinity purification strategy overall. And on other occasions, your fusion partner will start sticking to your target protein after you cleave them. So that's another problem. Now there are of course ways to address these problems during construct design in purification, but one thing to consider would be to leave the tag on, and this again depends on the application of your target protein. And that is, if you can still derive biologically relevant data from using tagged soluble protein, you should consider that approach instead of going through the motions of trying to cleave off the tag and then struggling to keep your target in solution. So in my earlier webinar in the summer, in response to a question, I had recommended that people who work in a core facility or for people whose daily work involves making soluble protein, it would be worth their time to acquire or generate a suite of fusion partner based vectors. And I just want to elaborate on that a little bit here through this schematic. For example, you have 4 different fusion partner vectors, right? Let's say GST, NuSA, Thioredoxin, and MBP. So you clone your target into all 4 of them at the same time and perform expression studies. So let's say one of them remains insoluble, so you discard it from further testing and move on with the other 3, right? You could purify them, but find that the proteolytic cleavage is an issue for one of them, so you discard that and move on with the other 2. So both of those cleave, but one of them precipitates out a solution post cleavage, but the other yields pure soluble protein. So now you have one good construct that you can work with, and that's the advantage of trying a parallel approach using multiple constructs at once. So here are some broad recommendations that I can make regarding cloning, expression, and purification. To increase throughput, excuse me, to increase throughput, it might be useful to adopt a ligation-independent cloning or recombination-based cloning strategy. If possible, use expression vectors with N-terminal solubility partners, for easy cloning of all targets. If you don't see target expression with an N-terminal construct, try a C-terminal one instead. That can help sometimes. Also note that if you are checking for enzyme activity, one version might show higher activity than the other. And also a C-terminally tagged construct will allow you to purify full-length protein. And you could also try a double tagging approach by using a solubility partner at the N-terminus and an affinity tag at the C-terminus to solubilize and purify full-length protein at the same time. So to summarize, there is no one simple global solution to address the insolubility issues in E. coli expression, but a well-engineered construct and a carefully crafted expression strategy can give you the desired results. Fusion partners can make a difference. Parallel approaches increase the chances of success. An approach that we can suggest is combinatorial tagging at the N-terminus. So basically a his tag and a solubilizing tag, followed by protease cleavage site for tag removal after the first round purification. And this schematic here illustrates a broad strategy. And finally, importantly, while solubility may be the most important element of a protein production campaign, it's not always about solubility, right? Expression levels, activity, purity, protein purity, homogeneity, protein stability, these are all important factors that must be taken into consideration also. And with that, before we head on to the Q&A session, I'd like to quickly tell you about our protein services that can help accelerate your research. We offer recombinant protein services in a choice of 4 expression systems: bacteria, insect, mammalian, and yeast. So in that sense, we are really versatile. In fact, we're one of the few service providers that have all 4 expression system choices. We offer a variety of purity and tag options. Many of our important services are comparable or better value than our competitors. Very importantly, gene synthesis is included in most of our packages at no additional cost. Many service providers will tell you one price, but when you get the quote, there are all these hidden charges that pop up. We don't do that. All our prices are completely transparent. Protein refolding, if required, is included at no additional cost. Many of our packages are guaranteed. So it's risk-free and no cost to you if we don't deliver as promised. We have the industry's fastest protein service. We have high throughput capabilities and and top-notch technical support should you ever need it. And this slide here showcases our experience and capacity in producing and delivering high-quality soluble recombinant proteins to customers around the world. Our production team has experience in purifying a variety of difficult proteins, including transmembrane proteins, cytokines, kinases, viral envelope proteins, etc. You name it, and we made it. And with that, we conclude the presentation and can move on to the Q&A. I hope you found this session useful. Thank you all for joining. Now, if you just give me a moment here, I just want to, before we go to the Q&A, I just want to say that this slide here lists some of our recently concluded webinars that you can view. Also, as I mentioned at the beginning of this presentation, this webinar will also be archived on our website, so please visit our webinar page to take advantage of these resources. If you have any questions or feedback for me, you can reach out to me directly via email. My email ID is listed on the top of the slide here. Also, it'd be great if you could please take a moment to complete a quick poll right after this webinar to tell us how you like this presentation. Your candid feedback will help us to improve our services. So let me see if any questions came in as I was talking. If you can just give me a minute here, I'm going to scroll now through these questions and do my best to answer as many as I can. Is it possible to predict the optimal expression system for a given protein sequence? Now, that's a good question. I'm not sure if we can predict an optimal expression system. This has to be done empirically, right? So obviously, many people start off with E. coli, just because it's easy to use, and once E. coli fails, they move on to another expression system. But if you look at your sequence. What I would recommend is look at literature references. If there is a precedent, if somebody has tried this in insect expression system and worked, and if somebody says that E. coli we have tried, it hasn't worked, mammalian hasn't worked, insect works, or maybe yeast works. So one way is to go about figuring out from publicly available sources. If it's a novel protein, novel gene, right, there is no literature precedent, this has to be done empirically. But then again, there are protein prediction programs out there that will tell you, for example, in E. coli, the likelihood of your protein being soluble. For example, there is a program that was developed by the Oklahoma University, or the University of Oklahoma, one of those. Now this program tells you the probability that your protein will be soluble when you overexpress in E. coli. So it's a really nice program that typically uses parameters like approximate charge average and the number of turn forming residues, and incorporates all this into an algorithm, and gives you an output, and gives you a customized value. Now, while this value is just a mathematical number, and it does not guarantee or indicate solubility, it's a decent and fairly easy exercise to perform, and it will give you some idea of your protein's expected behavior. Regarding the protein production service, do you supply the expression construct and protocol with the purified protein to allow one to prepare protein in their own lab in the future? Now that's a good question. Again, this depends upon the service that you have subscribed to. Now, some of our services we do offer. We do provide along with the pure protein, we provide the synthetic gene in Buck57, a COA report, a thorough report, and the QC data. Now those are the standard deliverables. Now typically if you need a protocol, that again depends upon, as I said, what service you subscribe to. Sometimes there can be a protocol transfer fee, on other times there isn't. So again, it's important to know that there are differences. So once you request a quote, our technical account manager can tell you if there are any associated costs with protocol transfer. How long should the linker site B before the protease cleavage site? Again, a good question there. Now the thing is, again, this is determined empirically. Typically the linker, I have tried in the past linkers that were just 4 amino acids long. Sometimes it did not work, so I've gone on to 8 and even 12. And typically I've tried before my protease cleavage site. If that doesn't work, I try after. But just to be on the safe side, I try and engineer a linker both before and after, because I don't want to go through the motions of trying one, trying to cleave, it doesn't cleave, and then engineer a whole new construct and try all over again. So my recommendation would be to play it safe. Try engineering a longer linker, because the probability of cleavage is higher with a longer linker as opposed to with a shorter linker. So I would say between 8 and 12 amino acids. So in case you're wondering what kind of linker, typically a combination of glycine and serine works really well. It's worked really well for me, and if you look at published sources, there are all sorts of linkers people have tried and they've worked really well. But from my experience, a combination of glycine and serine seems to work really well. Again, I have mostly used TEV. I don't like thrombin as much. TEV has worked really well. So about 8 amino acids before TEV, another 4 or 8 after TEV seems to work really well. What else? What about chaperone co-expression? You didn't talk about that. Yeah, absolutely. Chaperone co-expression is a way to improve protein folding in vivo. And now this talk was meant exclusively for fusion partner based approach, and so I didn't go into the co-expression strategy. A few E. coli chaperones have really been characterized well, like the GRO ES-EL pair and DNA K DNA J group E combination. So they work by promoting the proper isomerization by interacting with folding intermediates during protein synthesis. By the way, on this note, you probably also know about foldases like DSP-A, DSP-C, PDI. They carry out various functions, but essentially they all work to accelerate any rate limiting steps along the folding process. And while co-expression works well, it's important to optimize the strategy just like with anything else, because not all chaperones work well with all targets. What else? Also note that some chaperones can work really well as fusion partners. So I think I'm running out of time. I have time for one last question, and then we can wrap up. So why do proteins precipitate out of solution upon cleavage? Now it's really difficult to pinpoint the exact reasons, but solubility tags such as MBP and USE, they act as passive partners in the folding of target proteins, and solubility of aggregation prone targets after removal would primarily depend on the characteristics of the target protein itself rather than the tag used. So in other words, while the tag might make the protein more soluble, its role in causing the target to precipitate after you cleave it may be negligible. So I hope that made sense. So I hope I was able to answer all questions to satisfaction. And as much as I'd like to answer more questions, because of restraints on time, we'll have to wrap this session up now. I just want to let you know that we do appreciate your participation and all your thoughtful questions. Again, thank you very much for all your time and kind attention, and on behalf of the entire team at GenScript, we wish you all success in your research. Thanks very much.